Welcome back, deep divers. Ready for another adventure? Today we're shrinking down to the microscopic level to explore the world of microbiology.
Oh yeah, microbiology. Talk about an unseen world.
Right. We'll be focusing on infections and how we can figure out what's causing them. It's kind of like a medical mystery where we use science as our detective work.
Oh, I like that. A medical mystery.
And to help us illustrate this, we'll be using a fictional case study.
Sounds intriguing.
So picture this. We've got Cindy. She's a summer camp counselor who gets a scrape on her leg while playing basketball. You know, just your average playground injury.
Happens all the time.
At first, she thinks nothing of it. But then things take a turn. The scrape doesn't heal like it normally would, and it even starts to resemble an insect bite as it gets worse.
That's when alarm bells should start ringing. Our bodies are pretty good at signaling when something's off. And when an injury doesn't heal as expected, that's often a sign that an infection might be developing.
So did Cindy seek medical attention?
Thankfully, she did. The camp had a nurse who took a look at the wound and noticed something called pus.
Pus? Isn't that a clear sign of an infection?
It definitely is. So the nurse decided to send a sample from the wound to a lab for analysis.
So this is where the real detective work begins. We're moving from a visible wound to the invisible world of bacteria. Now what can we learn just by looking at bacteria under a microscope?
Well, different bacteria have their own unique characteristics, kind of like how we all look different. They come in various shapes and sizes, and they even have preferred ways of grouping together. Some are loners, some form chains, and others cluster together.
So it's kind of like their own unique fingerprints, and we can use those characteristics to identify them.
You got it. Just by observing these visual clues, we can begin to narrow down the possibilities.
This is fascinating. It's like we're about to zoom into a microscopic crime scene. I can't wait to see what they find in Cindy's sample.
Let's find out. So now imagine the lab that received Cindy's sample. It wasn't some fancy high-tech facility with all the bells and whistles.
Oh, really? Not quite CSI Miami.
Not exactly, no, but this actually highlights the incredible skill and resourcefulness of the lab technicians working there. Even with limited resources, they can still obtain a wealth of information.
That's pretty remarkable. It really speaks to the power of scientific knowledge and ingenuity. So how did they approach Cindy's sample with these limited tools?
Well, they began with a basic technique known as a wet mount.
Okay, I'm vaguely familiar with that.
It involves placing a drop of the sample suspended in water onto a glass slide and then covering it with a thin cover slip before viewing it under a microscope.
Yeah, I remember doing that in high school biology. But I have a feeling it's not as simple when you're dealing with bacteria.
You're absolutely right. Bacteria are notoriously tricky to visualize. They're practically transparent, so viewing them under a bright field microscope can be a real challenge.
It's like trying to find a needle in a haystack.
Exactly. But the lab technician had a trick up their sleeve. They decided to use a technique called dark field microscopy.
Dark field microscopy. Tell me more.
So instead of shining light directly through the sample, dark field microscopy utilizes a special condenser that blocks the direct light illuminating the background while leaving the bacteria brightly lit against a dark background.
Oh, that's clever. It's like trying to see stars at night. They're so much easier to spot against a dark sky.
Exactly. And using this technique, the lab technician was able to observe a key characteristic of the bacteria in Cindy's sample.
What did they see?
The bacteria were spherical and clustered together in formations that resemble bunches of grapes.
Okay, that's a visual I won't soon forget. But why is that shape and grouping pattern so important?
Different types of bacteria have their own signature shape and grouping preferences. Some are rod-shaped, some are spiral-shaped, and as for grouping, some prefer to fly solo, while others, like the ones in Cindy's sample, form these characteristic clusters. This information acts like puzzle pieces, helping us to narrow down the possibilities.
So it's like a bacterial lineup where we can eliminate suspects based on their appearance. This is getting exciting.
We're getting closer to identifying the culprit. To confirm their suspicions, the lab decided to perform a gram stain.
The gram stain. I remember hearing about that. What exactly is it again?
It's a staining method that helps distinguish between bacteria based on the structure of their cell walls. You see, bacteria can have either a thick, sturdy cell wall or a thin, delicate one. The gram stain basically highlights this difference.
So what happened when they performed the gram stain on Cindy's sample?
Well, when they viewed the stained sample under the microscope, they saw that those same spherical bacteria, clustered like grapes, were now stained a deep purple color.
And that purple color means?
That these are gram-positive bacteria. Their thick cell walls retain the purple dye during the staining process.
So now we know these bacteria are spherical? They cluster together and they're gram-positive. Those are some valuable clues. Are we ready to unmask the culprit?
Almost. By combining these visual clues with other tests and their knowledge about common bacterial suspects, we can finally solve this medical mystery.
Deep divers, we're back. It's time to crack this case wide open. We've got all the evidence laid out in front of us. Spherical, gram-positive bacteria, those grape-like clusters. What's the verdict?
Well, putting all the pieces together, this particular combination strongly suggests that we're dealing with Staphylococcus aureus, or S. aureus for short.
Staphylococcus aureus. I've definitely heard of that before.
It's a pretty common bacteria, actually, often found on our skin and in our noses. Usually it hangs out without causing any trouble, but it's known to take advantage of cuts and scrapes to cause an infection.
So it's kind of like an opportunist.
That's a good way to put it. And these S. aureus infections can range from minor skin irritations to more serious complications if they aren't properly addressed.
Oh, wow. So what happens next? How do we make sure Cindy gets the right treatment?
Well, identifying the culprit is a crucial first step, but the next challenge is figuring out which antibiotic will be effective against this specific strain.
I see. Makes sense. Not all weapons are created equal.
Right, so the lab will test Cindy's bacterial sample against a variety of antibiotics to see which one is most effective at stopping its growth.
So it's like a microscopic battle to find the antibiotic champion.
Exactly. And in Cindy's case, they did find a winner. The lab identified an effective antibiotic and her doctor prescribed it right away. But they also stressed the importance of completing the full course of antibiotics.
Even if she starts feeling better before finishing all the pills.
Even then, it can be tempting to stop taking antibiotics once the symptoms go away. But that could allow some of the tougher bacteria to survive. And those survivors can multiply, leading to a recurring infection. Or even worse, they can contribute to the rise of antibiotic resistance.
That's a scary thought. Antibiotic resistance is a big deal, right? Like those superbugs we hear about.
Absolutely. It's a serious public health concern that's making it harder to treat infections. Taking the full course of antibiotics as prescribed is crucial in preventing the development of these superbugs.
So Cindy's case really highlights how far we've come in understanding this microscopic world. We began with a mysterious wound. And using scientific detective work, we were able to pinpoint the exact bacteria causing the infection and find a way to fight back.
It really is remarkable. When you think about it, just a century ago, we were pretty much in the dark about these tiny battles happening inside our bodies. Now we have these incredible tools to identify these microscopic invaders and fight back.
It's mind-blowing. And while we've made huge strides in combating infectious diseases, as we discussed, the battle against antibiotic resistance continues.
Absolutely. Cindy's story serves as a reminder of the responsibility we all share in using antibiotics wisely and appropriately. Our actions today can have a big impact on the effectiveness of these life-saving drugs for future generations.
Wow, that's a powerful message. So deep divers, next time you hear about bacteria or antibiotics, think back to Cindy's story. It's a fascinating glimpse into this unseen world that's constantly at work within us.
Knowledge is power. The more we learn about the microscopic world around us, the better prepared we'll be to protect ourselves and future generations. Thanks for joining us on this deep dive.